Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
The iconic homunculus map, showing how the human body is represented in the brain, is clearly male. The drawing, published in 1950, depicts a penis deep within the medial wall of the cortex. Female anatomy, including the breasts, vagina, and clitoris, is omitted. The study also finds that the size of the brain region that activates in response to clitoral stimulation correlates with self-reported frequency of sexual intercourse. The paper is a step toward understanding something unique about the female brain, says cognitive neuroscientist Gillian Einstein of the University of Toronto, who was not involved in the study. The map showed that adjacent areas in the body are for the most part represented in adjacent areas in the brain, that is, somatotopically. The genitals were an exception, as they were found below the brain region representing sensation in the foot, in the medial cortex, along the deep groove that marks the division between the brain's two hemispheres. Several studies have investigated where the genitalia map within the cortex, using self- or partner-delivered stimulation or electrical stimulation to try to validate the non-somatotopic location of male genitalia next to the foot and to see if female genitalia map to the same location. In the present study, Christine Heim and colleagues at Charité Universitätsmedizin Berlin in Germany devised a technique to stimulate the clitoris without eliciting sexual arousal. We developed a technique to deliver an airburst via a membrane that allows for a precise stimulation while at the same time avoiding any discomfort, writes Heim, lead author of the study, in an email to the scientist. Importantly, the precise location within this region varied from woman to woman, Heim points out. But across all the women, there were no activations in other areas of the somatosensory cortex, beyond the region by the hips and upper legs. This observation is in line with a previous study in males using a non-arousing stimulus, which similarly placed the penis representation field at a somatotopic location between the legs and the trunk. As no size is specified for the membrane used to deliver stimulation, and the disc was attached above underwear, Komisarek questions how the researchers could avoid stimulating not only the clitoris's pudendal nerve, but also other nerves in the groin region. As a result, he postulates that the cortical near-surface activation is most likely due to stimulation of the groin region in addition to that specifically of the genitals. In an email to the scientist, the authors counter that a surface area of no more than 2 centimeters was stimulated, and they write, "We made considerable extra effort to develop a technique that is very precise and at the same time as minimally intrusive as possible." Komisarek also questions why the authors didn't analyze activation of deeper cortical regions. The problem with their claim is that they report only cortical surface activity in their study, ignoring deeper cortical regions. He writes, while his study and others report a deep cortical representation, especially of the genitals, in addition to near-surface representation. No area was left out in the 2D plane, including regions highlighted in previous studies by Komisarek and others. For Einstein, the precise location of the genital field matters less than the observation that the genital representation varies person to person. As far as she's aware, this paper is the first to look at individual variation in the somatotopic maps, she tells the scientist. She also credits the study authors for using a stimulus that did not lead to arousal, as sexual excitation may recruit more areas than those that would otherwise register sensation. In a second component of the study, Heim and colleagues measured the thickness of each participant's genital field by gauging the individual region that was activated the most by clitoral stimulation. Very interestingly, says Einstein, the results show a link between the two. In previous work, Heim and colleagues associated experiences of sexual abuse with reduced cortical thickness. These studies are very important for understanding not just sexual dysfunction as it might be described in biomedical literature, but also how people respond to sexual abuse, says Einstein. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.